Okay, so first of all, welcome to this CellProfiler workshop. Okay, it's going to be very interactive, right? So I will try to not say much through the slides, but it should be an interactive session, right? But also we have to take into consideration that it's just for 2 hours, so it's a really small time, especially for the software like CellProfiler, which is not as user friendly as Imaris and other professional software. So there are so many empirical values that you have to understand and different modules and stuff like that. But I will try my best to explain you, okay? And you can always raise your hand and ask questions in between. Okay. So have you got all of you the codes and the pipeline, which I said it to this one? This. Okay. Okay. That's the main issue, right? So if you go to this website, right, and you could download. So they say that Java installation is required because for the CellProfiler, Java runtime environment prior to installation. Mm, yeah, normally it still should work, but it's the best way is that uninstall it and then do it the procedure, install it again. Yeah, exactly. Yeah, uh, that's right. But don't you have this one, the what do you call edurom? So because you just have to install it, right? But that doesn't matter because you just need a internet access, right? Ah, okay, okay, okay. Yeah. Because normally I just put this website, the link, and I thought it should be okay, right? Because I think it's quite, yeah, okay. But anyway, so if you go to this Windows, right, from this Java runtime environment, you go to this one, accept license, yeah. Uh, yeah, for me, what works best is this one. Okay, so offline, Windows 64 offline exe. Did you try everyone this one? Because this is very straightforward. You just have to install it and install, hit the double click that exe file. So it will run in the background. It didn't work? Okay. Okay. You have to tell me personally what's the problem because this is maybe the system specific. Okay. But for me it worked well. Okay. But anyways, so we can't do anything for now. Okay, so I will just go on with my flow. But this is how you should do. So Java runtime environment, doesn't matter it's 151 or 152. You go to this offline version exe. Don't download this tarz. Okay, once you have installed this, go back and install, download this Windows version and install it. It's just very easy, right? It's just exe file. Okay, and so once you have installed it, it is very user friendly. See, they have given the user getting started. So CellProfiler is a free and open source software, and they have manuals, examples, and all other pipelines, which is useful for the biologist, right? They also have an examples here, basic pipelines for the human cells, fruit flies, all biological applications, right? And then they also have the specialized pipelines. Okay. And they also have specific toolboxes. For example, cell tracking, worm toolbox. Um, okay. They also have a forum. So it's very easy. You just have to sign up first. So if you click here, you just have to sign up here. And then once you have signed up, you're allowed to pose the questions. Okay. And it's very effective and very efficient, right? So you get a reply within a day. So they are very active communities. Um, okay. So this is just all the front end. Okay, this is also very important, the educational modules. So I like this one. So if you go to the educational modules link, and if you go to the video lessons here, educational modules, and this link, yeah. So she is Anna Carpenter. She is the director of imaging platform. She's the founder of the software, basically CellProfiler. But she explained all the methodology of the high content screenings, image based assays for drug discoveries and all. So it's very good actually. She has given a lucid explanation about from the very basics to the drug discovery, right? So I would recommend you to go there. Okay. Um, so then that's it. That's it for the web, how you install it and all. Um, so I'll just close this. Okay. So once you have installed, you will have this desktop icon here, CellProfiler. They have changed the image of the CellProfiler. So if you have an old CellProfiler, you will see the difference because this is the latest one. So you just double click. It will take some time. So this window will show you all the dashboard basically. It will show you what is happening in the background. So you don't have to worry about that. That will always be open once you double click. But it will take at least 30 seconds. That depends on how powerful is your computer, right? But it takes some time. Yes. Yeah. So once this is open, there will be three windows open. One, as I said, this is a dashboard. You don't have to worry, just minimize it. Second window will be just a welcome message from CellProfiler community. So so if you are a very newcomer, then of course they gives you okay how to load and how to download. This is what we're gonna see today, right? So I'll just close this. This is just the information basically, and this is the main window where you're gonna work most of the time, right? This is the front end of CellProfiler, and as you can see that this has been divided into different windows. So let's go back to the presentations. So before that, there is a disclaimer. So all the slides and the data set and the pipelines I have taken from the Broad Institute. Okay, it is already available in the public domain, so there is no, but I'm just giving a disclaimer for this. And all this presentation, this is the link for the presentation. Okay, right. So you must have heard CellProfiler and CellProfiler Analyst. What is CellProfiler? CellProfiler is nothing but the image analysis and quantification software. So you process your images, you quantify, you get a numerical data. So basically from image to numbers, that is done by CellProfiler. Once you have a number and images, the CellProfiler analyst will give you a data visualization toolbox. Okay, so that's step one is CellProfiler, step two is CellProfiler analyst. Yeah, okay. They both are open source and free. Right. What is the basic overview of CellProfiler? So you basically process large sets of images. This is the advantage of using CellProfiler. We can identify and measure the objects, and we can export the data as you can depict it in this picture. So we have this image. We segment the nuclei, right? And then we dilate this nuclei in order to get the cell boundary. And then we can extract different features, right? Once we have the masks basically of the nuclei and the cell, we can keep that mask on the original channel and extract number of informations which we're going to see. So the goal is to provide a powerful image analysis methods with a user-friendly interface. This is the whole goal of the CellProfiler, right? So if you compare all other open source softwares such as Fiji, they are not, I mean, they are very good, right? But you have to do a lot of coding, right? So you have to write a macros or plugins, right? And they are not tuned for the cell analysis, like image-based analysis. So there is no concept of cell primary object or secondary object, right? It goes through very basic level of image processing, right? Without considering the biological aspects of the cell analysis, cell-based analysis. And here the philosophy is you measure everything. You don't have to pose a question first, right? Which feature I want to quantify from the images. You extract all the features, intensity, shape, morphology, and later you decide, because we are living in the digital world with the big data, right? We don't have to worry about the space. So we extract everything and later we ask question, which feature is interesting for my readout. Okay. So this is the front end of the CellProfiler. By the way, this is the old version CellProfiler, but it's not different than the recent one. But what you're going to see is this this panel is the module. It's called as the input modules, right? Where you write all your modules. This is basically a set of instructions you write, And for so it has been divided into input modules and the analysis module. So input module will always be there by default. So whenever you open CellProfiler, input module will always be there. Okay, and that includes images, metadata, names and types and groups. We're going to see it now. And in analysis module, you write your instructions, how you want to process your images, right? Okay, the third window is the module help, right? So you can, for every module, you can actually ask help. What does that module indicates and what is that signifies? You can actually add and remove models using this plus and minus, and you can change the orders, right? Because this is just the algorithm, right? You're just asking an instruction. And for every module, when you click this module here, the property window will appear. So all the properties and the parameters for that module will be shown here. And the user has to just tune these parameters until he gets the desired output. Okay, so that's it. And if you see this input categories, the module categories, they have been divided into file processing, which makes sense, right? Because that's for input IO. You have to read images, write images, that comes under file processing. Then we have image processing, because once we have read an image, we have to process that image before you segment it. Very basic step. And then object processing. This is basically an image segmentation procedure, right? This is where you will spend a lot of time actually in the object processing. So once you have segmented your region of interest that you are interested in, then the fourth step is measurement. You put that mask onto the images and then extract the information. And once you have all the measurement, then it's a data tool, like how do you want to save it, right? And then there are specialized toolboxes such as co-localization, worm toolbox, and cell tracking and stuff like that. Okay. There is also a concept of image set here. Okay. So CellProfiler understand the image sets. What do you mean by image set? Image set is nothing but the multiplication, or it's a combination of different grayscale levels or grayscale images which has some similarity. For example, in this case, I have a DNA, which is a channel 1, and the GFE channel 2. This becomes one image set, right? Because it's a one biological entity, right? Even if they have a different channels. So for example, if you have a time-lapse data, all the time point images will be in one image set, because that's like a one, like it belongs to one entity, right? The reason why they say image set, because mostly this software is for the high content screening, and as you know, in high content screening you have a different wells, different positions. So different positions and wells, they becomes one image set. So every position becomes a one image set. Okay, so this is very important to know. This is just a okay. And before I go to this object identification, let's start from this one. So this is the front end basically, as I said before. And when you open, this is by default. Okay, this is the input module. Okay, the first step you have to do is, if you go to this the link that I sent you, and if you download all the data set and the images, you will see so many examples. The best example to start with is, let's go to scanner. So for example, in CPR we do a lot of image analysis using microscope, which is the high content microscope. And the images would look like this one, you see. So A01 is the name of the well, W is the well number, so W1 is a well number 1 and W2 is well number 2. P stands for position and Z stands for Z, right? So here it's just a 2D, so Z is only 0. T0, there is no time point here, and this is the channel information. Okay, so 53 BP1 DAPI and EDU and H2X. So I have 4 channels actually. So this would become one image set. Is everything okay so far? Yeah, just ask a question if you're not understanding. Yeah, right. Yeah. Not for the whole things, yes. Okay, so once you have images like this one, so you have your images here, and I have my algorithm, which is already written, but we will see what it does. Yes, all kind of formats. So it support, because it supports the bio format, which is it will work as if we are working on Fiji. So whatever the format the Fiji can support, the CellProfiler can also support that. And if it is not supporting, then it's always a good idea to just import all your data into Fiji and then export it as this format, which C stands for channel, T stands for time, Z stands for Z slice, the normal traditional format. Yeah, 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 exactly. Time. Yeah, yeah, yeah. So it's very important. Even though they claim that if you save this file as a one file, LSM or TIFF or whatever, right, just one file, sometimes it doesn't work uh, because of the metadata corruption, right? So it's always good idea to save one grayscale at a time. So if you have multiple time point, multiple channels, doesn't matter, or multiple Z, but as long as you have a keyword, let's say Z, C, and T, then it's fine. Then there is a way you tell CellProfiler how you want to combine this all data. Okay, we're going to see that now, because right, if you can see now, every one image is basically one entity. It's one channel, one time point, one Z, and one well, right, and one position. So how you proceed, you just drag and drop your whole folder here, and you can see here quickly all your images are there, right? Okay, so this is just for visualization, okay, that whether or not your images are there or not in that location. And you can actually scroll down and see, okay, there are some files which are basically auto generated, right, from the Windows thumbs.db. Don't worry about that because they are not image file, right? And why we are saying this, show files excluded by filters, and the filter is image only. The reason why we are saying this image only, because we do not want these guys, this thumbs.db to be included, right, because they are not images. So this is how you kind of exclude this images, the outlier, I would say. The second module is a metadata. Metadata is important when you have some kind of information within the file itself, right? So you can say yes, and once you say yes, you can actually ask, okay, extract from the file image header. So if you have a LSM file, for example, it will directly extract that metadata from the LSM file, okay? But most of the time it fails, you know, even though they claim, and I understand why, because their software has to update it with the bioformat and all these things. So that's why I said the best way is to import your data, LSM or TIFF or what, the vendor specific data, to Fiji, and then you go to Fiji and then make a composite images and then export it as a traditional ZCT format. Okay. um, yeah, just ask me if you have any question. Right. Okay. ZCD format is Z for Z stacks, C for channel, T for time. Okay, so for example, it's just that we do not have time, but I'll just show you now. So if you go to Fiji, I have it open here. And if you open, let's say, samples, there is a mitosis stuff. Yeah, 5D. It's a 5 dimension, meaning that you have a Z, time, and different multiple channels. You see? So you have a C, Z, and T, right? Right. So if you open all your LSM and TF file like this, and if you go here, image, stack to hyperstack, you have to mention how many channels you have. You have a 2 channels here. There are 5 Z slice and there are 51 time points. So as long as you have your data in this format, we say okay. You go to file and you say save as, but image sequence. Image sequence. Here it will write one file at a time, grayscale images. Okay, with this tag, I will show you that. You see here, T, Z, and C, and likewise it will write all your images. This is what you should do always. close it. Maybe in that case, I think I would not confuse you. Maybe we should take the example of because I can see now it would be difficult for you to understand. So I'm just taking this example, translocation 2. Okay. Okay. So let's say you are in this window. You don't have to close this window. Okay. If you want to, let's say, process another data, you can always say new project and say no. So everything will be rub off. So it'll start from scratch. So I'm just drag and drop this image, and then this one here. Right. So if you see my input folder, this is the images, and in this images also A01 is a well ID, right? S1 is a position, S is a position here, and W1 is a wavelength, which is the channel one. So there are two channels only here, but there are multiple wells. Okay. Right. So once you have uploaded this data, you will see this. If you want to visualize how the image look like, then just right click, show selected image. This window will pop up, right? Just to ensure whether the data is right or wrong, right? You can zoom in with this symbol here. You can zoom in, go back to the home condition. So you can pan basically. Well, after zooming, that makes sense. You can pan it, right, left, whatever, right? And that's it. Okay, so I just close this. This is how you visualize it. Okay. The metadata, right? Because we have to tell now that what is the structure of my files, right? What is the structure of my files here in the metadata? So if I go to CellProfiler, just go here. Right. So what is the structure? So I say extract metadata, and I say yes, right? Because I want to extract the metadata from this one because there's individual files, right? And I say extract from file and folder names. Okay, because I want to extract from the file names. Because if I say extract from the file headers, meaning that when I have LSM or TIFF file as an individual file, it will automatically extract from that header, which I know by the experience that will never work, or most of the time it actually doesn't work. So that's why we are asking from files and the folder names. Okay, so from file and folder names. And metadata source file name, because we have written for every images the name of the file contains some information like ZCT, right? This is what I'm saying, the file name now. And this is a regular expression. Okay, we can spend hours on this one, how to write a regular expression, because it's basically technical stuff, right? But what you are telling is we are making a rule now saying that the first keyword is my plate, which we have seen actually it's A01. What was the structure? The structure is this one. See BBB C013 A01. So first of all, before this A01, this is common for everyone. Okay, so this is just this data is from one plate. This is what means, and the plate name is basically this BBB C013. But how to tell CellProfiler that this is my plate, and this is what I'm telling now. So yeah, this is just a regular expression. Don't worry about that. But yeah, this is what we have to create. So you have to click here, and then you have to create here. You have to write accordingly, right? So what I'm saying is that, okay, take everything which is at the start and everybody and consider that as a plate. So that's my plate ID. Second one, underscore. If you see this image, right? So there is an underscore here, underscore. I'm putting an underscore here, and then say whatever comes after underscore is my well actually, right? But I have to define how this well is represented. and that well is represented from A to P. So that character can go from A to P, right? And it can take from after this character there is a number, which is true, right? Because 0 1 here. So I'm saying that it's from 0 to 9, but it has a 2 decimal places. This is what I'm saying here with 2. Okay, and then again the underscore and S. S stands for position, so I would say site. By the way, you can write your own coding here, so I can say position here also. That's my choice, what I want to call. Okay, also the site goes from 0 to 9, which is true, S1 2 3 4 5 whatever, and then underscore W and then channel number, and that channel number can go from 0 to 9 because we have only 2 channels, but doesn't matter, right? So, and you can see once this is right, this regular expression, it will not give you any error, and it will highlight all the places. So basically red one is the plate, this one is well, this is position, this is my channel. That's it. Yeah. So you're asking about this one. So this is my choice, what I want to call that. Okay, so I can say here now, for example, I can say it's CPR channel, whatever, right? It's my choice basically, what I call. But I'll be shown the CPR channels likewise. CPR channel 1, CPR channel 2, like that. Okay. Yes. Yeah, yeah, yeah. Yes. Yeah, syntax. Yeah, this syntax, like for example, question mark P is very standard for CellProfiler. It will always start from question mark P and then in the arrow bracket, and then you say write whatever the name you want to write. That's it. And important is this keyword. This has to be consistent to the what the data you have, and you can get what? Yeah, I mean for you offline. Yeah, yeah, I'm recording this one, and this already there actually. The star says that take all the plates from the data. See, because we are calling from the folder, right? And within the one folder we have multiple images, and every images with their name has some information. And CellProfiler is asking, you want to extract this metadata from the file name? Yes, this is what we're saying now. Okay, we say submit, right? And to see now, so extract metadata from all images, of course, you can say update. Once you say update, it will show you all the information. You see here, plate now, it is the same for everybody because there is nothing else. Site is from 1 to, this is only one site. There's a well, right? There are different wells here. And the channel, you see CPR channel now, it has changed. 1 2 3 4. It is just like a name that we are giving to the channel, right? Because I have changed now, so I will just go back and I will just drag and drop. You see, this is the rule set that I have already written, right? So because I have changed the CPR channel name, everything else needs to be changed. So that's why I'm just dragging drop again this thing here. It is asking me, do you want to overwrite? Yes, I say yes. Now it goes back to the original one. So if you go back to metadata now, see, this is the channel number. Got it? Yeah. Okay, once you have extracted the metadata, there is the third module called as the names and types. Okay, because we still haven't told that, okay, W1 and W2 is my channels, but we haven't called them, right? And this is that third module will say. So in the third module, names and types, image matching the rules, yes. Process as 3D. Does your data set is 3D? Do you want to segment 3D data set? But we know that this is just a 2D data, so we say no. That's why we are here. I have one more example for 3D, then that time I will explain you. Select the rule criteria to match. Then I say, okay, for all of them of the following rules, because we want the file does contain W2. Give that, so whatever the file which has a W2 should be given the name raw DNA. Okay. Make sense? So you can do like this. So you extract from the metadata. Okay, you go to the metadata. Actually, because it didn't upload it properly, but you go to metadata and you say now here does, and then say have channel number matching. See, and then you say here how many channels we have basically here. So it goes from 1 to 2, right? So it goes from 1 to 2. So I would say just 1, right? That is my DNA. And what kind of images I have? That's a grayscale image I have, right? Okay. How about the other one? Select the rule criteria match, because I have 2 channels, right? Then again I go back to metadata and I say does have a channel matching 2. So that is my raw GFP. Okay. So I call my first channel as a raw DNA and second channel as a raw GFP. Uh, this one doesn't appear, this thing, everything below this. Okay, let's do one thing. I just say remove this image, and so by default, so if what happens with names and types? Do you see this one, this window here? Okay, so this is fine until this point. Okay, then you click on this one, duplicate this image. So yeah, so duplicate this image, go to metadata again, and then change it to 2. And here you say it's raw GFP basically. Okay. And you can always say update, right? Just to check. And you can see now the raw DNA and raw GFP, and this becomes a one image set, as I said, because there are 2 channels, that becomes one image set, right? So, oh yeah, go ahead. Yes, yeah, yes. I mean, you don't have to say time, because time, if, you don't have to say time. Let's say if you have a 100 time points, you don't have to say it 100 times, because right now we want information, we want different processing for different channels. That's why we are making this. You're understanding the difference? You don't process differently for different time points. The reason why we're making this, because we want to call 2 different channels, because we want to do a segmentation separately for one channel and the other one. That's why we're making this data set. So the first step is just images. Metadata is just how you call, how do you want to call that data set. And then images and types, meaning that how do you want to stratify that data set. Okay. Again, the groups, this is not important, because in the groups, let's say for example, you have a 96 well plate. You have positive control and negative control. So you can also make a group of that, right? So here I would say, okay, take it from, let's say, well, and based on well, I want to make it as, so say remove this data set, and if you go back and forth, and if you come back again, you see here now. So every well, so I want to stratify my whole data set based on the wells. This is what I'm making here now. Do you want to group your images? Yes. Based on what? Well. Yeah. And you can, how many wells are there? There are 4 wells. No, there are actually, yeah, these many wells. But some of them are actually fictitious, because in the metadata we put actually from A to P, and even if in the data set there are not enough images. So here don't worry about that. Normally I never use this group images, because once you have all the information, you have one of the column as a well. Later you can always segregate it, so you don't have to do it necessarily here. So I would always say no here, we don't need that. Okay, now this is all about the input data. Okay, so we have basically extracted the metadata, we have called, that's channel 1 and channel 2, which is DNA and GFP. And now it's a question of, what is your question now? The first step is that you want to segment the data set now, segment the nuclei, which is the DNA now. So for that, you just right click, add, and go to object processing and say identify primary object. Yeah, you don't have to, because right now, because I do not want to. Okay, what I can do is actually I just delete everything. I just delete everything. So you go shift, click here, everything will be deleted now. So this point is okay up until this point. Okay, then you just right click, object processing, identify primary object. Can you see this now? Okay. Once you have this, you say the first question is use advanced settings. So for time being, we say, okay, we do not want to use advanced setting. Select the input image. You remember we created two images, raw DNA and raw GFP. It is asking me on which channel you want to apply. You want to make an object from. So I would say DNA, right? Okay, how do you want to call that? I would call that as a nuclei, right? It's nuclei. The typical diameter of the object. What is the approximate diameter of your DNA, the nuclei? To check that, you go back to images again, right click. So you see, we switched the channel now, because our channel 1, which is the W1, is the GFP here. So we have to go back, names and types, and here I would say 2, and here I would say 1. You understand? Right. So I go to here now. This is my nuclei. To check what is the approximate size of the nuclei, you go to tools, measure length. First I would zoom in a little bit just to see the approximate. Tools, measure length, the approximate diameter. The length will appear here, display here. That's around, you see this the bottom corner, so it's around 12. Here, tools, measure length. Okay, and then click here again. That's around 12. Okay, so close this, go back to this algorithm again, and I have to give the tolerance, like the range basically. So that's around 10 to 40. So nuclei can go from 10 to 40 size. Okay, discard object outside the diameter range. Yes, right. So if you want, if there is a nuclei which is outside this range, that will be discarded. It won't be segmented. Discard object touching the border of the images. Yes, because we do not want the half nuclei, right? And that's it. So let's see the result of this one, right? Because this is the segmentation algorithm. To see the, so you can actually run in two ways. You can either just process all the data that you have using this setting, but right now, because we are setting the parameters, so we are not sure about the parameters now. So it is always a good idea to go with the test mode. So we can always come back and forth. So you will see these two tabs here. One is analysis images, meaning that you want to just analyze all the images. You are very confident about the settings. But right now we are not, so we just say start test mode. So if you click on start test mode, right, then these two tabs will appear. One is a run and one is a step. When you click on run, meaning that it will just run through all the modules. But we do not want to do that, because we want to go step by step. We want to check the output of one module, then second one and third one. So that's why we say step here. So if I click on step, the output will be shown. You see output of the nuclei. So this is my input image, this is my nuclei, and this is just the boundary of the nuclei. And it's showing me there are 306 nuclei that it found under the criteria that we actually set it. Okay, and you can actually zoom in and see the effect. I think it has done a very good job, even if we are not doing an advanced setting. Okay, this is just the basic algorithm. Everything fine? That's fine. Good. Okay, so once you have done this, we have given name to nuclei. What is our second option? What was the second thing that we wanted to do? We wanted to do with this GFP. If you go back to the images, this is my GFP. I want to make a cytoplasm, right? That is one criteria. But to make a cytoplasm, uh, I will start with the nuclei as a seed point, and I will start growing on that channel, on this GFP channel, right? Are you following this part? Yeah. Okay. So once you have made a primary object, because we are starting from the very basics, so I would say at this instant you can start asking and the different measures, different features from this primary object itself as a nuclei. Okay. So you can just say right click and say add, go to measurement, and you say object intensity. You can also right click, go add measurement, go to size and shape, for example. So all my morphological information and the intensity information will be there, right? So with this with the measure object intensity, you have to mention select an image to measure from which you want to extract this intensity, right? Nuclei is my object, but from which channel? So I have to say, okay, I want it from DNA, because let's say I'm interested in DNA intensity of the nuclei. And also I can add actually the another one. I can say, okay, I'm also interested in GFP intensity. Which object do you want? Of course, I want nuclei. You see, there is a lot of combination, permutation combination. You have a two channels and you have a two object. So far, we have just segmented one object, which is nuclei. So there is a two option of extracting the intensity information, one from each channel. Right? This is what I'm saying now here. Similarly, for this size and shape, I have to mention which one, which object basically, nuclei object, right? And it's also calculated Zernike features. That's basically advanced morphological information. Okay, so nuclei is we gave the name nuclei to the masked object, the mask object. Yeah, yeah. That's what it's showing me this one. Because if you go back now here in identify, and if you change this name, that name will appear the next, right? That's quite obvious. Mhm. Yes. So when I say measure object intensity, it is asking me from which channel. See, because intensity is a imaged specific information, so you have to give on which channel. I have a choice because I have two channels in my data set, image set. So one is the DNA and one is the GFP. Now here you have to ask your biological question. Like, does it make sense to get this information from the GFP channel or DNA? It depends, right? Maybe if you want to see whether or not there is a cell proliferation, G1 phase, G2 phase, then I need integrated intensity of DAPI, right? Mm. Yeah, exactly. And also the DNA content, because we have two images here. See, two images and one object. So there is a two option of getting measurement, right? You can also add another one, but we haven't created anything else. Okay. So this is what you get, right? And before going to this complex issues of creating cell wall, I will just finish this how to export this data. Let's assume that my biological question was, what is the integrated DNA intensities, DAPI intensity within this hole? And then I want to see it from the well to well, how it behaves. So once I have done this, I just right click, add, and say measurement. But I want to now export it, right? So I will go to the data tools, mm, file processing, just a minute. Export something, export to spreadsheet. Yeah, file processing, export to spreadsheet. And if you click here, export to spreadsheet, everything else will remain same. You have to give where you want to export this data, right? So I can by default it will go to this output folder. This location you can change it from here. So view output setting, you can change this location. By default it will go to C users and your user ID, right? But let's say if I do not want to save it here, I will go here and say elsewhere, click here, and I can have my specified location. So CellProfiler. What example we were doing here now? It's a speckle, right? No, doesn't matter. I'll just make a raw output or something. I just say OP here. Select folder. Add prefix to file. Do you want to put a prefix to the file? I would say yes. You can give whatever the name that you want to give, right? Experiment number. Overwrite without warning. No, no, no. I think you don't have to change anything here. And you say export all measurement type. You say yes. That's it. And the export to spreadsheet will not work in the debug mode, meaning that you have to analyze it. It will not work with the debug mode. So to get rid of this now the test mode, you just say exit test mode, because now we are confident, right? Our setting is good. So we just click here. So it will process all your data set, all your images, and just to see. Okay, and before that also, there is eye open and close. So let's say if you want to see the intermediate output, then you can keep your eye open. This is what it means actually. You understand? No. Okay. So for every module, there is an output, right? There is input from the previous module and there is an output for that module. If you wish to see the output for every iteration, then you can keep this eye open. So what you're saying is that I would like to see for every images the output of this identify primary object. So the problem with that would be whole your desktop will be populated with many windows. Of course, yeah, right. Normally you do this only with the debug mode when you want to optimize your parameters. Once you know that the parameters are optimized, why do you want to see it, right? It doesn't make sense. So that's why I would close all the eyes. Is that fine? And then just hit on this analyze images. Now it will call all your parallel computing toolboxes. So it will use all your cores. That's the good part of CellProfiler. So it doesn't use only one core, but it uses multiple cores. So it's fast actually. Okay. So I would say analyze images. The check mark that will, yeah, this check mark would just like a sanity check by the CellProfiler. You cannot do it. It says that everything is okay. This is what means. Yeah, it means that you have to change some settings. There is some mistake. You haven't selected a particular module or something. If you put a cursor over there, it will give you a warning that what is a mistake that you did. What does it say in if you put your cursor on that? Okay. I think just for the sake of time, maybe we can just discuss this offline. Okay, your personal. Okay. But you see now, it's using 8 workers, meaning that I'm using 8 cores of my computer now. Yeah, go ahead. Yes, you can either do that or right click and say disable module. Yeah, you can always disable. Right. So all this option comes when you develop a rule set where you're not confident, right? Which rule set will work and what. So you can always keep your eye open and close and stuff like that. But once you're confident, it is always a good idea just to hide that or delete that module, right? You don't need that. Um, so it says that 0 of 26 image set. So if you work on the powerful workstation, this will be done in 2 minutes. But because my laptop is recording and then doing a lot of other things, that's why it's slow. Okay. So I'll stop now because now you understand what would be the output, right? It will be an Excel as fine. It has asked us which format do you want. It's a comma separated by default, right? So I would say stop analysis. And this window will pop up once everything is done. It's asking us open default output folder, save the project, right? Or say okay if we do not want to do anything. Nothing has been written because we have yeah, stopped this processing in between, so nothing has written. um, this is good. Okay. Mm, right. So here, now let's look at the very interesting one. So here there is an images, as I said before. There is a BP1 channel. BP1 is responsible for mitotic damage, right? It's a marker for mitotic DNA damage. And I have a DAPI, and I have different wells. So A01, A02, A03, and A04. So I have 4 wells. I have 2 channels, DAPI BP1. I have one position only, P5, only one Z, only one T0, and well is nothing but A01. So see, so A01 is the W1, A02 is W2, and likewise W4, right? Because you will have this situation, right? Whenever you have a data, you will always have a grayscale images, and the naming convention of that grayscale images should have the information. Then only you can assemble them and make as an image set. Okay. So now our job is, now we have to segment the DAPI, and for every DAPI make that mask onto 53 BP1, because BP1 only appears in nuclei, right? It's a nuclei marker basically. So we have to make that mask and put it on the BP1 channel. But also in BP1 we are not interested in the BP1 integrated intensity. BP1 has like a foci-like structure. If you see here the BP1, if you open, you see there are foci here. So our interest is to count how many foci are there within the nucleus, right? And we want to see how many, what is the distribution of the number of foci per nuclei from well 1 to well 4. That's our ultimate goal, biological goal. Okay. So let's start from the beginning. So again, before that, if I want to save, there is a 2 ways of saving. One is I can save this entire project. You see, entire project save as. So what it does is it saves this rule set, my algorithm, all the modules, and also the data set. Not like a raw data set, but the location of the data set. So next time, if you just open that project and if you hit this button, analyze images, you will get an output. Okay. Or maybe if you do not want to save your data set, but let's say you are just building this pipeline as a generic pipeline to apply it for the multiple replicates, then you can just go here, file, and say export as a pipeline. Okay, export as a pipeline, and then that's it. That pipeline you can just yeah, drag and drop again. So I'm not going to save this now. I would say new project, so everything will be go off. You see, so by default these modules will always be there because this is the basic ingredients, right? This is the basics, yeah, steps for the cell profiler. Plate, just drag and drop. So I have all information here. It is in JPEG format. It's not a TIFF format as before, so doesn't matter which format you have. So first I have to assemble it, right, in a correct order. So I would go and say, okay, this format. So this example is scanner like. Okay, there is a folder called as scanner like, and there is a folder which has images, and this is algorithm. Okay, so if you are at the first step, everything is fine. See here what I'm doing here now. Maybe because for the sake of time, I do not want to process all the 4 wells. I just want to go for the positive control and negative control. First, I want to see whether or not my controls are good. then only I will go for other siRNAs screening stuff. And just for the example, I took W1 as my positive control, W2 is my negative control. Okay, and I just want to see the output of only these two wells to see the effect, right? So this is what I do. So I just close, for example, show files excluded by filters. So I say yes, I want to exclude custom, right? And say any. So select the rule criteria match any and say file does contain. So I just want those file which contains only W1 from all these files name. So because if you see the file name, it contains W1 to W4. I only want only W1 and W2. Okay, so what happens now, and I say apply filter to the file list, you see everything is deactivated now, everything else except W2 and W1. Good, right? This is step one. Metadata, again, because we have to tell with a regular expression how you want to call it, right? Because if you see the images, it's a different keyword for that. This is what I'm telling. So if you go here, this is, so I don't care about A01 here, because as long as I have only one indicator for well, that should be enough. So I'm not asking anything for this A01. I don't care what that A0 or A02 is, but I'm starting from hyphen W, and then anything after W should be considered as a well, right? So this is what I'm representing as a well, that the number can go from 0 to 96. In this case, it's only 2, we know that, but that doesn't matter. And then what is the maximum decimal places that they can go? So from 1 to 96 is a 2 decimal place, like from 1 to 2. So that's why 1, either it can be 1 or it can be 2 decimal places. Again, hyphen P, and whatever after P will be considered as a position. The number can go from 1 to 8, because we know already that there are only 8 positions that I have scanned within one. Digits, exactly. I would say the places, basically the placeholder. Yeah, if let's say 1 and then 12. Yeah, exactly. Yes, yeah. I mean to say the places, basically. Yeah. Okay. So as you can see, the P, it's 1 to 8, so of course it is 1 only, right? It is not like 10 position. So in that case, I should have given 1 comma 2 decimals. Okay, again hyphen Z0, because Z0 and T0, they are common, right? There is no multiple Z stack, there is no multiple time points, so it doesn't matter to mention anything about that. And then hyphen channel, because there is no keyword for that. Anything after this hyphen should be considered as a channel name. This is what I'm saying now here. And then dot JPEG. The star is because consider everything after that. That doesn't matter, right? As long as you mentioned this one. Sorry? Yeah, it's a wildcard. Exactly. Perfect. Yeah. I just do not want to mention this technical term, but I'm very happy that you know that. You're aware of this. Okay, just say submit. So all images. Here is a very interesting thing, right? Because now I am actually convoluting all the information. So I have my plate information from the folder. Because see, the reason I'm showing all this thing, because this is a part of a big screen that we are doing now. So we have basically 112 plates for I think it's around 1000 siRNAs. Now I think 1000 genes and then 3000 siRNAs, right? The 3 siRNAs targeting. So we put basically all images into one folder and call that folder as a plate 1, plate 2, plate 3, likewise plate 112. So I want to show also the plate ID, but I want to show that plate ID, take that information, extract from the file and the folder names, but this time not file name but the folder name. You see, because if you go here, image, what is the folder name? It's a plate 1. So I'm just giving that information also in addition to whatever the information I have here. Yeah. Okay. I know it's a bit complex, but once you are aware of this one, it's very easy then, right? You just have to understand this. Okay, again the metadata, and I'm saying that folder name, and I'm saying the plate underscore, that's a common keyword, and anything after plate underscore should be considered as a plate ID. This is what I'm saying here now. Okay, you can say update, and you see here that there's a plate 1. That's plate 1 position. Well, everything is fine. Once you are happy with that, then you can go ahead. Then, but we still haven't mentioned the channel now. This is what it comes now. Names and types here. Names and types. Again, image matching rules. Is this a 3D data? No, it's only 2D data. Then it's no. Select the rule criteria. Match all. Yes. Metadata does have CPR channel matching DAPI. You understand this part? Because if you go here, there is a DAPI channel here. This, and we mentioned actually that whatever is the last after hyphen is a channel. And you can see it from here, right? This is output. What is the channel CPR? There are only 2 options here. 4 options. Yeah, but we are only interested in DAPI and 53BP1. That's why in the names and types, we are just creating DAPI and 53BP1. If you want EDU information, which is a cell proliferation marker, I can also go and say, okay, duplicate this image, and I want to call it BP1 is done, does channel which has EDU, for example. It should be the same as the name that I call, and I can say EDU here. But if I am not going to process this channel anyways, there is no point in creating this channel, right? Because my interest is only 53BP1 foci count per nuclei. So I would say remove this image. And also I could have gone for the gamma H2X, which is also DNA damage marker, right? Okay. Important here is that I'm calling this image the channel name, but I cannot call which that starts from the digit. The channel name has to be called with certain, yeah, it has to be a character. It cannot be a digit. So I'm just calling that a 53BP1 here. That's very important. But it's just a limitation from the CellProfiler. They do not allow the channel name to be start from the numerical values. That's it. That's why I'm just calling here underscore. Okay, and the type of this image is a grayscale. That's what I'm saying, grayscale here. Okay. So we have basically done everything now. Do we want to group? Yes, because you remember that we selected only 2 wells, and one is a positive control, another is a negative control. So I say, okay, based on well, yeah, do that. So there are only 2 counts, right? For group 1 and group 2. Because I think there are 2 positions we taken, right? Okay, so this gray to color, that is not important. So not to get confused, right? So we have done everything now. What was our first goal? Our goal was to segment the DAPI now, right? So right click, add, object processing, identify primary object. This is what I have done here. Identify primary object. Okay. Last time we have seen without advanced setting, but this time we'll see with advanced settings. Okay. So when you say advanced setting, select the input image. This is very obvious, right? Because it's asking on which image you want to create DAPI nuclei. I would say, yeah, DAPI. What do you want to call that? I want to call that as a nuclei. What is the typical diameter of that object? How do you get this information? You get this information from go to image, DAPI, right click, show image, and just get an approximate size of DAPI. Tools, measure length, go here. That's around 80, right? This is showing here. So I would say go back, identify prime object. So it can go from 40 to 200, let's say. Yeah. Okay. Discard object outside diameter range. Yes, we do not want anything else. Discard object touching the border of image. Yes. Threshold strategy. Here there's a lot of okay, I have to spend a lot of time on this one because this is a bit complex, this part. And this is the reason why you don't have to do a lot of programming and the macros, but they have taken very good care of this one, like a best logical combination one can have to have the desirable output, right? Okay, threshold strategy, you have 2 options, global or adaptive. What do you mean by global? So I will go back to the PowerPoint here. this one. Let's say you have this image, okay? This image, doesn't matter which, this image, nuclei, right? And you know the histogram of the any image. So first, the basic step of segmentation is to first plot an histogram. This would be the histogram. What is histogram? The x-axis is the pixel values, and y-axis is the how many times that pixel has that particular value. So if you take any image, the majority of the pixels will be background pixel, right? And that's why you see this peak here. This is one peak. And the second peak is most probably your object of interest. But this is not always the case. There will always little bit small bumps over here. It means that your object of interest is not homogeneous. There is a lot of intensities, which is not background, but in the foreground there is a lot of variations. Okay. So one way of finding this threshold, and everybody knows what is segmentation, right? I think I have covered. Segmentation is like basically finding a threshold. After that, I mean, more than the, if the pixel intensity whose intensity is more than that threshold would be considered as your object, and anything below will be considered as a background. Right? That's the segmentation. So you are creating a mask basically. So if I put my threshold here, which is an obvious case here, then it's good, right? And that can be done automatically. That algorithm called as a Otsu algorithm. Okay, Otsu algorithm. But before you find this threshold, one can do global, meaning that I want to find an histogram of the entire image, okay, and based on that I want to find a threshold strategy. That strategy is called as a global, meaning that you are taking entire image histogram into consideration. But if you just go and make a window which is proportional to the size of your object, and then making local windows and then finding a threshold locally, then it's called as a local threshold strategy, which is basically it's asking us global or adaptive. Okay, so most of the time global will work, most of the time. Yes, yeah, exactly. And you might want to consider this. Let's say you know a priori that there is an uneven illumination within the cell itself, you know, which is very unlikely to happen in high content screening, but we never know. If this is the case, for example, if cells are one GFP positives and you want to only focus on, if you want to segment only the cells which are GFP positives, then their intensity is maybe uneven. In that case, you might want to consider this approach of adaptive. Of course, it takes longer. Yeah, I would say when we are talking about CellProfiler, it's better to forget 3D, right? Because it crashes most of the time. Yeah, I think Imaris is the best one, I would say. If you have a 2D data, then I would consider a CellProfiler because this release, this is the first time they are actually released as a 3D data processing, right? So they are not stable. It is not yet stable. I think it will take a couple of years, I would say, to reach to the level of Imaris and the Velocity and other commercial software for 3D perspective. 2D is really good. Yeah, it is comparable to Fiji and other open source softwares. Even I would say Scanner, which is commercial. Yeah. So this is a global. I have one example 3D. If you have a time, I'll just show you. And then we have to go back to Imaris just to visualize the data because you can't visualize here. We can't visualize 3D data. Okay. Then my strategy is global here. And once I know that, okay, take the histogram from the entire image, then it is asking me different algorithm for segmentation, finding a threshold. You see this part here, um, this one. This finding of threshold will be done by different algorithms, and the basic algorithm is Otsu, which is showing here. That's Otsu here. But again, Otsu, it is asking, do you want 2 class or 3 class? You know, 2 class is basically, if you go here, there is only one threshold, meaning that background and foreground. But if you have a 2 classes, meaning that there will be two threshold, so that it will divide into three regions. And it is asking me then for 2 class it doesn't ask anything, but if you go for the 3 classes, It will ask me assign the pixels in the middle intensity class to foreground or background. That's my choice. How do you want to assign the middle section of the intensity? Do you want to assign them as a background or foreground? I think you would appreciate now the way they are thinking. You know, because it's very unlikely, uh, most of the time you do not have a very good signal to noise ratio, right? Between, let's say, bright nuclei and everything else is black. In that case, one threshold should be enough. But maybe in some cases you have a faint kind of a boundary, like intensities outside your object of interest. So in that case, you segment that part, also background, but that part also which is kind of a faint, not bright, but also not dim, and also the brightest part. And once you have this third class, middle class, you assign that middle class as a background. So everything will be then nicely bright spot segmented. You see? I think you have to see the result, right? Back and forth. And that's why they have given this test mode, right? So you can, let's do that actually, because, so foreground, let's go to 2 class, okay? And again, let me explain this part and then we'll go back and forth with the different settings. So with the 2 class, threshold smoothing scale. Sometime what happens that if you go here, smoothing, this one. Even if you apply, this is my object of interest that I want to segment. But because you see there is a 2 maxima here, because the DNA contents are not homogeneous within the nuclei, but computer use intensity information. That's why it finds this 2 and it finds and basically cut it into 2. But if you smooth it, this image, before you apply the threshold, we will get rid of this problem. This becomes 1. Smoothing, you know, everybody is smoothing, right? If you smooth an image with a Gaussian kernel, this is what that kernel size is, 4 or 8. If you go beyond, then it becomes a very blub, like a blurry image. But there is a problem also. It won't work all the time, because if you blur too much, smooth too much, this problem would come. This problem, because if you blur too much, this would become one object then. You see, because there is a 2 nuclei here, but because you blurred too much, the boundary of this nuclei and that nuclei merge together, and that's why it becomes 1. So it's not good also. So you have to make a compromise, right? That's a problem. But this is what that factor is, threshold smoothing scale. And 1.34 meaning that you want to increase that threshold. So whatever the threshold that you have to smooth that threshold, smooth that image basically. This is the kernel size. This is the kernel size, 1.34 meaning that 1.34 pixel. Okay. I think it's too much technicality now, because if you want to convolve your image with a Gaussian, you need a Gaussian kernel size, and this is what that size is basically. But for you just keep on increasing this number, the image becomes smoother and smoother, right? Threshold correction factor. Remember that you find this threshold, that value, but maybe if you want to be very sure about that, I want to find the very positive contender for that threshold, then you can multiply that threshold value to the positive or negative. You can decrease or reduce that threshold, okay, by some factor. Normally you just have to say 1 here most of the time, but the reason why I'm telling you is because I think it's important for you. I'm pretty sure that most of the time you won't work, you would just work with the default values, but this is the significance of changing, right? So if you see, let's say if a nuclei, if you see over segmented, maybe there is a point of increasing this factor so that the over segmentation will be reduced and it becomes a manageable stuff, right? Okay. No, this is not erosion. Erosion comes when you create your object of interest. This is not erosion. Okay. Um, th this one was my here. Okay. Let's say my algorithm has found this threshold here, but you know that this threshold is not good enough, because this threshold is here, these pixels will also consider as a foreground, which I do not want. Then I have a possibility of shifting this threshold to the right side. so that the most bright object will be considered. Understand? So it's basically you're first finding this threshold automatically using two criteria, right? One is adaptive and a global. Within adaptive and global, you have a choice of which algorithm you want to find that threshold from. Once you have found the threshold, you have a possibility of smoothing your image before you apply that threshold, or you can scale this threshold with some factor. So there's a lot of variation here, lot of permutation combination, but it's really good because then, yeah, you will find some solution with a lot of combination with this one for your images. This, yeah, it no, so scaling meaning that it smooths the entire image before even it starts asking for the threshold also. So smoothing has nothing to do with the adaptive and uh, global. You can still the no. So this one, see, threshold smoothing scale. This you understood. Correct threshold, correct factor. This one is that, see, the output of this global or adaptive is an output, right? That, so you have a choice of multiplying that threshold with some factor. You can check that. Let's say, for example, if you go to adaptive, see, there's a correction factor. It's still there. Understand? So what will happen with this adaptive? For every nuclei, there is a window, right? Within that window, it will find a threshold. Let's say with Otsu algorithm, it has found some threshold. Now it is user, you would say, okay, I'm not happy with that threshold. I want to increase the threshold for that particular object. And it does it for every other object because you are on adaptive mode, right? But if you are in the global mode, it just does it this one time, scaling of this factor for everybody. Okay. Again, there is a lower and upper bound for the threshold. So I know it's for the beginners it makes a lot of complexity, but it has really a really good sense, right? Because if you try to optimize it, lower and upper bound. So the best example for this one, imagine that you have high content screening. You have taken many images, and in some of the wells, some of the positions, there is no cell, and because of the hardware focus or the software focus, there is a completely black image, but there is some uneven illumination because it tried to stage the dynamic range, right? Because you have told your algorithm to find a threshold anyways. That's because we haven't told which is what, which is object or which is not object, right? It just tries to find for every image some threshold and try to optimize. So for the, let's say if there is no nuclei, but there is a black image, so with this algorithm, it will find some threshold and it will try to segment some kind of a very weird structure, right? But that threshold value would be very small, right? Because there is, if you see the histogram, it won't be too much distance between the two distribution. They will be very close. In that case, you can set a lower bound for the threshold. I would say no matter if you find a threshold, but if that threshold is less than zero point 0.2, do not segment that image at all, meaning that that image itself is not segmentable. Okay, so this is what that lower threshold means. It's the same thing for the upper threshold also. Let's say you have a very bright structure, right? Maybe, I don't know. Normally in the radiology, it happens. If you have a metal stuff, you know, and if you try to make a CT and MRI, because of this metal dynamic range, the whole dynamic range of the image increases, and there is a big blob there, right? A bright spot. And if you want to get rid of that, you can also set a maximum upper bound also. So yeah, 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 exactly. Yes, yes, exactly. So basically, this is to get rid of the debris in your image, right? Okay. I think we should see now the output because we are just talking. um, okay, 0.0. Okay, let me finish this and then we'll see the output. What happens is, let's say you have done segmentation very well, everything is good, but the problem would come if the cells are very close together, if the nuclei are very close together, like what we see in us. Imaris, we do watershed basically. And everyone knows the importance of watershed, right? Because if you have images, this is called a clumping. If the cells are very close together, and if you apply threshold, this would become one object, right? So how to tell computer that these are different nuclei? So that is called as a declumping. This is called as a clump, and this is called as a declumping. The algorithm, there are so many algorithms. The one basic algorithm is a watershed, right? So what you do is basically you see this image as a profile with a crest and trough. This is a crest and trough, um, um, the peak and valley. And so peak is basically the maximas of intensities. And imagine that you are pouring water from top, and the water will not stay at the peak, right? It will go to the catchment basins. And this is how the computer comes to know wherever there is a catchment basins, I need to cut it there. That's like a terminology they use in computer science, right? But the point is that you find local maximas. Once you find a local maxima, then another question comes is that how do you want to cut that nuclei? Then there are two options in this. Either you can cut this based on intensity, meaning that you have this peaks intensity. Then I would know that okay, I have to draw a line over here, or based on shape. If the shape of the object is like that, then it does some mathematical morphological calculation, and it comes to know that this is the least or the strong gradient curvature, I would say. What are the points within the boundary where there is a strong curvature? I would say this is a strong curvature, and it tries to find a line which would separate these two objects. So this is based on shape, right? There's no intensity here. Otherwise, you can choose based on intensity. I think in nuclei it makes sense to have an intensity because there is a local maximus for every nuclei, right? But sometimes it doesn't help, right? Because let's say if your image is not a DNA nuclei, a DNA stained, but you have some GFP where it is distributed, it's not a high peak. But because your cell is like that, so GFP would be like this. If you see this, the segmented object of two cell, GFP would be like this. And then it's an obvious choice to use shape information here because I know this is a curvature where I need to cut. I'm not using intensity information, but I'm using shape information here now. Okay. Yes, that you can do it using the shape and size criteria, the filtering of the objects. Yeah, you can do that. It internally it uses. See, watershed is just the way of explaining to the lay. Yeah, exactly, the biology. So, but normally it uses this distance transform. So no matter if you use intensity or shape, it actually goes through this distance transform. Yeah. Um, yeah. Okay. I think we should see now the result. Um, okay, and this is what it means. So method to distinguish clump, yeah, based on shape or intensity. But again, there is one more point here. The point is that once you have done this segmentation, right, there is also a choice of drawing a line. Either I can draw a line based on the midway between the two intensity center or the center of the two object that I have segmented based on maybe shape or intensity, or I can draw this line where there is a demeshed line between these two objects. Yeah, I think biologically this makes sense to use it because yeah, the two nuclei cannot be exactly the midway, right? Okay, so according to this situation, you have to change this, right? Okay, now let's see the result. So to because we haven't done any processing, right? And we have to be on the test mode. So I would say start test mode because I'm not sure what is the setting is. So I would say start mode, test mode. And then here I would say I want to see this result. That's why I'm opening my eyes now here, right? And go to the step here. It takes time. this is the output. Okay, are we good with this? It does a very good job here. But see the effect of see here, this thing. I think it's the best person is to judge this validation is a biologist, right? Because as a computer scientist, we don't know what this biological entity is, right? Are they two or one? So yeah, if you're happy with that, that's fine. But let's go back and change some strategy. Let's say I go to identify primary object, and I would say, okay, instead of two class, I would go for three class, and I would take the middle class as a background, not foreground. So I want a very brightest intensity DAPI. Okay, and I would say just say step again. So see, you can go back and forth. You can optimize your parameter, come back and see the result again. So as you can see, it's not as fast as Imaris, right? Because this we are just processing only 2D image, one image, and it's taking that long. But of course, it's free, it's open source, so that's a good part. This is the result. And now what is the difference? Yeah, you would for sure see the brighter pixels, right? Because if you see this pixel here, see they are gone here, this region. If there had been only two Otsu, this would also come into picture, right? This is just an example that I'm giving you, but you got the point, right? So maybe this is the middle class, and that middle class we have given that to assign it to background. That's why it's not showing here as a foreground. Okay, but let's see what's the output if I change. Let's say I do not want to declump the object. You have to wait, right? You have to be very patient with this. Sometimes it crash also, and you have to start from the beginning. But as long as you're working on the workstation, that's fine. But don't work on your laptop. I mean, if you have a 2-3 images, that's fine, but not for a entire screen, for example. Yeah, or 3D images. Now see, it's not showing my text off here. Okay. So let's say I use intensity here and intensity. Let's see the result. So it's 121 this time. So it's lower, right? It's less than the previous one. I wouldn't go for the intensity. I would go for shape. You see, this is the reason. Because it's going by the intensity. This is what we selected now. Say intensity. So I would go for shape. That's more clever way of doing that. But again, it changes from data to data and application to application. Okay, this is the one part. Um, you can also see the result with adaptive filter, right? With adaptive, you have to mention what is the size of your adaptive window, right? What is the size of your nuclei, and that window will go iterate over the entire image and try to do this for every image. Of course, it's going to take a lot, but you understand this concept of adaptive, right? Okay. So if you have uneven illumination within the object, then use this adaptive. Uh, yeah, so let's go here and say mm, no adaptive, but the global. And if you smooth it, right? So smoothing threshold, let's do it like 2. It won't show us this image after smoothing, but it applies threshold on the smooth image, not the original image. This is what it means, right? And this threshold correction is that if I go for, let's say, 1.5 here, And let's put it as before. And if I put 1.5, see the output now, and everything is shaped. That's fine. So the brightest contender will come, the nuclei. So it has done that. Okay. And all this pink one is like it is discarded because we have set this size as a criteria. You remember that there is a range that we are given size. That's why it is discarded. Okay. I think we should move forward because then otherwise won't be finishing this. So let's say we are happy with the DAPI, right? Now the second thing is we want to do this BP1 foci, right? And if you see the image of the BP1 foci, if you just apply the segmentation thresholding, do you think it will work? Because see the quality of your image is not that good, right? It's very dim, the spots. So if you doesn't matter if you apply what strategy of thresholding, we will not get the optimal output of a foci-like structure. So to have the robust output, we have to pre-process this image. We have to enhance this feature of the spot-like structure, right? And this is what I'm doing in the next step. There is a tool module called as enhance. So image processing, and if you go to enhance or suppress the features, enhance or suppress the features. If we click that, I'm asking on this channel, of course it is asking me BP1 channel, and I want to give that new image name as a BP1 enhanced image. Okay, what do I want to do? I don't want to suppress, I want to enhance. What do I want to enhance? I want to enhance the speckle. In this case, speckle is the only option, right? Because others is like we don't care about that. In this case, the speckle. What is the feature size? Meaning that what is the approximate size of the speckle that I'm interested in, like BP1 foci. Again, go back to this measurement and then get that approximate size, right? So if I go here again, BP1, and then go to tools, measure, put here. What is the size? Around 8. So that's around 10 I would put, right? Because it's showing here now. Close it, go back, enhance, and I put it 10, right? That's it. Then I hit on next then. You see, I haven't, I did not open my eyes here, so I won't be able to see what's the result. So I would say, I want to see it actually. So you hit this one. But I think it would go. So in that case, I have to exit this test mode, right? You have to start from the beginning because you forgot to open the eyes. So I would open all the eyes by default, and then we can close later. Right? Test mode. So it will start from all the way from the beginning. So first it will identify primary object. We don't have to see now. It's fine. Enhance. This is the output of enhancement. I'll just close this. You see the result now. You got it? So all this background, which is the bigger than the 10 size, is completely subtracted, and it only enhances the speckle-like structure, which has approximate size of 10. and that would help us in segmenting, right? Because if we segment from this one, this blob will come as a one foci. Got it? This is what we're doing now. The algorithm is called as a rolling ball algorithm, and again it's a bit complex because we don't have time, but there is an algorithm for that. Okay, and that algorithm is called as a rolling ball algorithm. Let me see quickly if I have that. uh, this one. Normally I explain during the Imaris workshop. This is a rolling ball algorithm. So what it does is basically there are a lot of peaks here, right? These are as if there are the speckles, right? But these mountain, the bigger mountains, they are kind of a, a blob-like structure, the bigger blobs. On top of that, there are spikes, right? This is what the situation is if you see it in a line profile way, right? So we basically choose this ball, which is the radius that we chose, like 10, and we as if we are basically throwing a ball. Now, depending on the radius of the ball and the height of the peak, the ball either topple over or it will fall down like this one. You see? So it will not go down because the distance between the two peaks are the smaller than the diameter of the ball. You understand? And what you do is basically you get the profile of the ball, and you reduce this profile of the ball, subtract this from these whole peaks, and you get the spikes. That's the whole idea. Then basically then you can put one line here as a threshold, and you can segment all the spikes in one go. This is what it does. So we have done this. Then again we will segment, right? Then again the identify primary object, and I would say use advanced setting, but I want to use BP1 enhanced now, not this BP1, the original one, but the enhanced one. Yeah? And I want to give the name as a BP1 foci. That's my object. I have to give again the same thing, size criteria, 10 to 25, everything same, global, Otsu, 3 class. Again, I want to be very sure about the positive foci. That's why I'm giving middle class as a background. Yeah, 0.1 smoothing factor, 1.5. Let's see what happens, right? Very bad, right? There's a lot of over segmentation it is doing. So you have to go back and change your algorithm setting. How about this one? It has done a good job here, right? But not here, not here. So maybe you just go back and change your settings. So maybe in this case I would go for adaptive, right? Because globally doesn't make sense. So I'd go for adaptive, and then here I have to choose a window size. So I would put it as 20 and see what happens next. Okay, we just quickly will go through this one. If not, you understand the flow, right? We don't want to optimize. We don't have time to optimize this now. See, it's taking longer because it's adaptive, right? Still bad, I guess. See, a lot of structures, false positives are coming. And in that case, maybe what we should do now, we would increase this smoothing. So just put it as a 1.4 or something. So we want to smooth this image first and then try to apply. Correction factor 1 point, let's say 2. Let's see what happens, and maybe not. Sorry? Yeah. So see, there are a lot of permutation combination, right? So what I'm trying to do now is I'm smoothing now, and I'm putting the increasing the threshold. Yeah, the bound is when only when the threshold is calculated, right? If that calculated threshold is below than 0, in this case it won't consider, or if it is above than 1, then it won't consider. That's why the bound is. But I think first we have to optimize before that. So let's see what happens now, because I think it should work now, because we have smoothened our image. It was 0.1 before, so that you can just, because it's a plugin, right? You can write one plugin, and then after that it's your choice what to put in, right? In that case it's very flexible. You can use the plugin which is written by others also. In this case, here we are restricted by their logic, their way of thinking, right? So if, let's see, we good. No. Yeah, you see here, so we are kind of getting there. So this is big, but then I would just go for, I will increase some factor, right, of the intensity, and the rest I will get rid of that based on some filtering also. But at least we are getting what we want here. Of course it's not an optimal one, but yeah. So we have this BP1 object, BP1 foci. Now we haven't told that this BP1 foci belongs to a parent object, which is the nuclei. This is what we're telling now, relate object, right? So right click, add object processing, and you say relate objects, because we want to tell who is parent and who is child. And if you click on relate object, who is parent, so I will select nuclei, and the child object is BP1 foci. So every nuclei will make a cover, and we'll see how many foci are there within its umbrella. Right? Calculate per parent mean for all children measurement. Yes. So let's say if you want to quantify sum of BP1 foci per nuclei, or the sum of intensity of BP1 foci per nuclei, that also you can calculate. That will be the average for one nuclei. Okay. Calculate child parent distance, none. You have to say centroid if you want to find object-based colocalization. Let's say you have a 2 protein of your interest. You do this speckle enhancement and the segmentation, and then you want to find if how distant they are, their centroid is from. That you can see it here then. Okay, I would say relate object, then the measure object intensity. Okay, we have just given this relationship to the cell profiler, and we say measure object intensity. So select image to measure. So I would say BP1, right? Or I would say BP1 enhance in this case, because I do not want to have the background into consideration. So BP1 image and DAPI image, which object? BP1 foci and nuclei. Two options available. So there will be two by two option available. Two object, two channels. Gray to color, the reason why I'm saying gray to color is because I want to also show as an overline. If I want to report, if I want to publish the data or in the group meeting, then I want to show also the overlay of the segmentation on the original image. So for that, I'm just saying gray to color. So select a color scheme, so that's RGB. I'm saying select image, so that's the first one is DAPI, second one is BP1, and the third one I just want to leave as a blank because I have just two channels, and I want to give that as a name, so that's a color image, right? Okay, so but this is, I have just made that RGB channel using two channels. Then you say overlay outline, so right click, image processing, overlay outline. Okay, what I'm saying is that you outline the overlay using display the outlines as a blank images. No, color image. You see, this is why I made this color image, because I want to put overlay on the image that I have created, right? That's the color image that I have created. I have to give the name for that. That's outline, color, thick. You do not want inner outer, but thick line to the boundary, and select object to display. So of course I want to display nuclei with blue color as outline and BP1 foci as a red outline. Yeah, and save image. Again, it's in the data processing, right? Data tools and the save image. File processing should be here. Save images. I want to save that overlay also for every image. Okay, once you have done this, then what we did before also, export to spreadsheet. And here the comma separated, otherwise what options we have? Tab separated, two options we have. Tab separated, comma options. Where do you want to save it? Yeah, I want to save it here, and the same thing, and export all measurement. So now it will export, because we have given this relate object as a module, we will have one of the column which would have the mean information of the BP1 objects. So like how many BP1 objects are there per nuclei? What is the mean intensity of all the foci together for one nuclei? Like all such average information we have. So with this we can actually then average all the nuclei from one well, because you remember that we made two groups based on positive control and negative control. We can see the bar chart that within one well if the nuclei has more foci or not, like more DNA damage using this or not, kind of, right? So that's it. And once you have done this, by the way, you can also pause it, right? So when you are in this exit mode, sorry, the debug mode, you can pause it here, and you don't have to then do this step by step. You just click on this run button, the counter will come and stop here with the overlay outline, and later you can just go through. You can go ahead later. So if I now say run this, so it will not export to the spreadsheet because it will not work in the debug mode, but at least we can see the overlay. Okay, so let's see this overlay at least for one image. Save images. Where did I save? I would say elsewhere, and I would save it to C, CellProfiler, and OP here. Select the folder. I would say I pause it here and I'd say run. Okay, we should have closed our eyes, right? But now it's only running for one batch, right? So that's fine. But you can always go here and say, okay, Windows. Now I don't want to crash it, but Windows, and say hide all the windows, so it will hide. Okay, let's check our output folder. Yeah, there is one. See here? Because we have given blue, so at least we are getting what we want, right? I think next what we'll do, let's come to the very good conclusion before we stop. We can also, because we know there are a lot of false positives on our BP1 foci, right? What I will do, I will not, I will also measure the shape information of BP1. I want only BP1 foci whose shape is not completely haphazard like algae-like, but like a roundish shape. And based on that, I will filter, right? So for that, I would just go right-click, add object processing, and I would say first I have to quantify the shape information. So measurement, size and shape, right? And here I have to select on which BP1 foci, right? Calculate sanity? No, I do not want to calculate it. So go, I switch off this, and I come here, and then I say step, right? So I would now measure all the shape and size information. Yeah, see here? That's BP1 foci, and this is the all the morphology information, right? So mean of eccentricity is 0.8. Also, there is something called as a form factor. Yeah, 0.42. So if form factor is 0 means it's a circle, okay, and it's 1 is eccentricity. It's ellipsoidal. or it's other way around also, because that strategy changes from software to software. In MRS it's different, and here they have different, but there is a way you can evaluate the size and shape. So let's say eccentricity here now. Okay, we have calculated that. Now I would say, okay, after this calculation, add object processing, and I would say mm, filter objects. And in the filter object, I would say I want to filter BP1 foci, and I want to call as a filter object. And measurement, I would say, yeah, from the measurement, I want to give the limits and the area shape information, not the intensity location number, but the area shape. And within that, which feature I want to evaluate. Let's say form factor, filter using the minimum measurement value. So I can give, I want those whose form factor is at least minimum this one, kind of this one, yeah. And same for the maximum. So for maximum, I don't care. And minimum, let's say if I say 0.5 or no, 0.3 or something. Let's see what's the result after this. So switch off and I exit mode. See what's the result. It has to start with all the 7 modules, but there is a way you can then filter out, right, the foci. But they also have a cluster support, so it's not that slow is not the criteria. That's not like a disadvantage, I would say, because there are many institutes, they have taken commercially this cluster support, so they can put their data like cloud computing. It's also support cloud computing, so you just have to put your data and then they will process with the different nodes, multiple nodes. So in that case it's fast. Mm-hmm. Yeah, first you have to quantify that feature, and then you have to apply that with the filter. Yeah, here. Yeah. Yes. Yeah, yeah. But here there is no filter intensity. So in the filter object itself, we have to select which criteria. I have selected shape criteria. There was also option of intensity. I can also choose that, because that also makes sense, because a lot of false positives are very black, right? So I can check. So there is multiple ways, right? In MRS is just a slider, right? It's very fast. You select, but here it's also possible. It's not that interactive. So you can see here now. See 644 were there, and it's only 547. So yeah, so maybe this one. But maybe we have to use some other criteria then, right? Like eccentricity or form factor or whatever. The combination, maybe we can also do a combination. So if you go to filter object here, limits, area, shape, I have these many options to choose it with. So eccentricity, and eccentricity minimum, if I put 5 for example, let's see. No, it didn't work. Yeah, so you have to come up with some, yeah, you need a time for this. If I put 7, let's say, what happens? Yeah, 503 out of this one. But this is gone, you see. This one is gone, this remains the same. So I think we are doing the other way around, because if you filter out, I think this is what we have to set, because we want eccentricity. So small eccentricity we want. Maximum, it should not go beyond, let's say, 0.3 or something, and then it should come now. Because MATLAB has a different um, what do you call mm, the guidelines of eccentricity, the definition basically. It's a mathematical formula, right? So I always get confused. And this is, yeah, you see here, it's only one remains. That's like a very positive containers, and they're roundish in shape. This is what we want. Okay, finally, right? 11, which makes sense out of 600. Yeah, yeah, exactly. Yes. But with this overlay, we can do that, right? Because so what we have to do is we have to go, yeah, 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 yes. So we have to go back and now we have to change this model, right? So on that we can shift it, right? So for example, relate, from relate to control this thing. You just see this one. I just take it down, say all. So that will be yeah, shifted downward. And then if you're done, then just click on the start again. Then hopefully you'll get the desired output. Okay. I wanted to cover many more modules actually, but now I think we yeah, there are so many applications, right? Co-localization we have a tracking, but co-localization and tracking, I would always say go to Emaris. Especially if it is 2D, then it's fine. But if it's 3D, then really there is very complexity, right? Because maybe you have also have a time lapse data. You want to see how the nuclear bodies are actually co-localizing over time with a different cell phase. So if there is a lot of dynamics with the co-localization, yeah, and especially if we have that license, right? I mean, if you have Imaris, then I don't see the point of using, yeah, CellProfiler for that. Yeah, that's the best advantage. Yes. We don't have it, but we have it in CPR. But in DanStem, because batch processing, the way batch processing works in Imaris is that you have to have a images, right, with that. Normally in DanStem, we have a one big image, but we don't have many of such. In CPR, we have like 2D images and we have many. So in that case, batch makes sense. Yeah, yeah, right. No, yeah, yeah, right. But there is a, yeah, there is a one hack that one can do. So without having a batch license, you can make a fake time points. Okay, maybe I'm recording, I should not say this. You can make a fake time points. Okay, let's say you have many images. Let's say you have different whales and different positions. You make that as a fake time points, and that becomes your one image. And Imaris can read that at a time. There is no size for that, and you just have to build your algorithm once and let it apply for every time points, and later you make a lookup table that this time point belongs to this well, like that. That's without spending, yeah, 10,000. But that's very expensive. Euro, I think 6 7,000 euro for batch processor. Yeah, but do this. I mean, that's it. Yeah, and we have script ready for that, and because I have done it for when we did not have CPR, a batch processor, I did that. So the script is already ready. Yeah. Okay. Thank you so much. I hope it was helpful. Um, and I hope the recording was there. So you're welcome.